Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at how we classify viruses into different groups. And so one of the first questions we must ask ourselves is, why is there a need to classify viruses? And I guess just like our eukaryotic and our prokaryotic organisms, classification helps us to arrange various entities, including our viruses, into categories based on shared characteristics. Essentially, it provides a framework for studying the relationships between different viruses and their evolutionary history, all of which will assist in diagnosing and treating viral infections more effectively, whilst also informing strategies for disease prevention and vaccine development. And so historically, when we look at how we used to classify viruses, it was based on things like their structure, the type of host infected or the tissue they infected, how they're transmitted, and the type of disease they caused. However, as our technology and understanding of molecular biology advanced, so did our classification methods. And so today we classify viruses based on information such as their genetic material and how they replicate their genome. Now, one of the key classification systems we use in virology to this day is the Baltimore classification system, which was developed in the early 1970s by Nobel Prize winner David Baltimore, and essentially this system groups viruses based on the nature of their genomes, the mode of replication, and ultimately how they express their genetic information. Essentially, all viral replication cycles revolve around a central stage of messenger RNA synthesis. And this itself is dependent upon the structure of the genetic material, so whether it is DNA or RNA, whether it is single or double-stranded, its overall replication method, and whether it is positive or negative sense. Now, what do we mean by this? So as a quick recap, when we talk about nucleic acids, they can have different roles, and to help determine the role of the strand, we can classify them as being either positive sense or negative sense. And so positive sense strands are also known as coding strands, and they have sequences and codons that are directly readable by the host cell's translational machinery, helping to synthesize proteins. Essentially, it has a sequence that corresponds directly to the RNA transcript, meaning it can also be classed as the non-template strand, as it mirrors the template strand. And then on the flip side of this, we have negative sense strands, which are also known as template strands, with sequences and anticodons that are complementary to the RNA transcript, meaning it can also be classed as the antisense strand or the non-coding strand. And so the sense of a nucleic acid strand tells us if it can be directly used by the host cell machinery or that it needs to be processed before this can happen. And so this is especially important when we start to classify our viruses. And if we now look at how the Baltimore classification system actually works, viruses can be classified into one of 7 different groups or classes, and these classes are commonly referred to by the nature of the genome associated with that class. So first we have our DNA viruses, of which there are 2 different groups. Class 1 contain viruses which have a double-stranded DNA genome, with both strands being able to be transcribed in a similar fashion to that found in host cells. Class 2 also contain DNA viruses, however their genomes are only comprised of a single DNA strand, which can be either positive or negative sense. Next we move on to our RNA viruses, of which there are 3 different groups. Class 3 contains viruses which have an unusual double-stranded RNA genome, which the host cell treats in a similar manner to double-stranded DNA during transcription, the only difference being some of the virally encoded and packaged enzymes are required to initiate the transcription process. Class 4 and Class 5 viruses are single-stranded RNA viruses which are either positive or negative sense respectively. Both require a number of virally encoded and packaged proteins in order to replicate the genome. However, they both have different ways in which they are transcribed. Then, our final two classifications under the Baltimore system are what we call reverse transcribing viruses, or RT viruses. So for our class 6 RT viruses, they have a positive sense single-stranded RNA genome, which has a DNA intermediate in its replication cycle, hence the term reverse transcription, which can then be used for the generation of messenger RNA, and therefore be translated into viral proteins. Then our final class are the class 7 RT viruses, which have a double-stranded DNA structure and an RNA intermediate within the replication cycle. So this is almost the opposite of our class 6 viruses. And essentially, when we look at our RT viruses, they form intermediates before forming messenger RNA, and this distinguishes our class 6 and class 7 viruses from our class 4 and class 1 viruses respectively. And so by using this classification system, we are able to cluster viruses into groups depending on their genome and how it is converted into viral mRNA. Now, just like any system, it has both positives and negatives. And one of the core strengths of this system is that it's capable of grouping viruses based on fundamental unchanging characteristics. And once a virus has been assigned a class, we are able to make certain predictions on its molecular processes, such as how it replicates, how its genome is transcribed, and whether it requires virally encoded proteins for these processes. However, as with all models, they are never perfect, and I guess one of the main drawbacks of this classification system is that whilst viruses may be genetically similar in terms of genome structure, they can vary wildly in respect of their overall structure, meaning it can somewhat oversimplify complex aspects of virology, including their biology and interactions with host cells. However, on the balance of everything we know so far, it's been a really useful tool in the classification of viruses. Now finally, as you might already know, a mutation is where we get a minor change occurring in the genetic code. And because the genome of a virus is quite small, along with the fact that they don't have any inherent system to correct mutation effects, the result of even a single nucleotide change can have major effects on the virus. And depending on where the mutation occurs within the viral genome, it can affect the overall biology of the virus, making it more or less virulent, altering its transmissibility between cells, tissues and host organisms, and even affecting its evolution, generating phenotypic variants with their own evolutionary paths. Now, for the most part, these changes or mutations won't change the virus as a whole. They're not going to be big enough to change its Baltimore classification. However, it can alter its ability to cause infection and the severity of infection once initiated. Now, whilst any virus can undergo mutation, some viruses are more prone to mutations than others. So for instance, our double-stranded DNA viruses tend to be more stable with lower mutation rates compared to single-stranded DNA viruses, with our various RNA viruses being more susceptible to mutations again. And so through the various mutations of different viruses, we have arrived where we are today, with thousands of different viruses, hundreds of which infect humans, and some of which are able to cause debilitating disease, whilst others act as mild inconveniences. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.